us for the Southeast Ocean and Coastal Acidification Network State of the Science webinar series. Today we will be hosting a virtual panel entitled Science, Industry, Management, Perception of Ocean Acidification and Fisheries in the Carolinas. This series is hosted by the Southeast Ocean and Coastal Acidification Network, or SOCAN. In short, the series lays a foundation for the state of ocean acidification science in the Southeast region. These webinars are intended to create a dialogue among scientists and stakeholders to identify what is known, what isn't, and what research in other regions of the U.S. can be applied to better understand ocean acidification and its impacts in the Southeast U.S. With awareness of and access to the research and its applications and uses, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. My name is Libby Jewett. I'm Director of NOAA's Ocean Acidification Program, and I am a member of the SOCAN Steering Committee. I will be moderating today's session on behalf of the SOCAN Steering Committee. Jen Bennett-Mintz, Education and Outreach Coordinator for NOAA's Ocean Acidification Program, is also on the line and will be helping to facilitate the session. During the presentation, all audience members will be in listen-only mode. You are welcome to type questions related to technical issues or questions for the panelists in the bottom of the control panel on the right of your screen. We'll be monitoring incoming questions. After all panelists share their perspectives, we will move to questions from the moderator, and then we will take the questions from those online to continue the discussion. We are also recording this session. The video recording and PDF of the presentation will be available on the SOCAN website. We are very excited to have 4 panelists with us today representing the ocean acidification and fisheries sciences, natural resource management, and industry sectors. Although it would be great to highlight all of the work each of the panelists has been involved in, for the sake of time, the introductions will be very brief to allow more time for the experts to share their perspective and for discussion. You can find the panelists' full biographies on the webinar section of the SOCAN website. I will give a brief introduction before each panelist speaks. So without further ado, we're going to move into some short presentations from each of our panelists. Our first panelist is Eric Smith. He is Research Coordinator for the North Inlet Winyah Bay National Estuarine Research Reserve, and he will offer the ocean acidification science perspective. Eric is also a research assistant professor with the Baruch Institute for Marine and Coastal Science at the University of South Carolina. Sorry. So without further ado, I'll turn it over to Eric.
Thanks, Libby. I'm really glad to be part of today's discussion. I think it's an important topic. So to jump right in, my goal in this very quick introduction is to contribute 3 what I feel are key points to the discussion. First is that the process of ocean acidification is really a relatively simple fact of chemistry, and there's not much debate regarding the process itself. However, things are always more complicated in the coastal zone compared to the middle of the ocean. And while I think this makes life interesting, in this context, it hampers our predictions regarding future pH levels in the coastal waters specifically, because we also have to account for possible future changes in the other drivers that control coastal CO2 dynamics. And here I'm specifically thinking of eutrophication, nutrient enrichment, and its effects on biology. So just to make sure we are all on the same page, the conventional definition of ocean acidification is the physical dissolution of increasing atmospheric CO2 in seawater. The chemistry is pretty straightforward. The more CO2 there is in the atmosphere, the more CO2 will dissolve into the marine waters. This will react with water, form carbonic acid, increasing protons, which is our definition of decreasing pH, and ultimately decreasing carbonate ions, which is what the critters need to form calcium carbonate shells. Like I said, the chemistry is straightforward. It's well understood, and in fact, the process is directly observable in the open ocean today. As an example of that, I'm showing a time series from Nick Bates et al. From the Sargasso Sea. This is a 25-year record of observations right off of Bermuda at the BAT station. In the top panel, you can see a very clear increasing trend of CO2 in seawater that roughly parallels increasing atmospheric concentrations, which is the pink squiggly line. As an almost mirror opposite, there's a steady decrease. These are seasonally detrended data, I should say. A steady decrease in pH, along with a decrease in aragonite saturation, which is related to carbonate ion availability. How these trends will continue in the future really depends on what future CO2 emissions do. To date, anthropogenic emissions have lowered oceanic pH by about 1/10 of a pH unit. Predictions for our future CO2 emissions are sort of all over the map, but they suggest anywhere from an additional 1/10 to maybe on the order of 3 or 4/10 of a pH unit by the end of the century, 2100. I think the consensus is pretty clear that this is happening, it's observable. The predictions, while they're based on what future CO2 emissions do, are not in a tremendous amount of dispute. I think the issue really is when we get into the coastal zone and talk about ocean acidification in estuaries in the near shore. In addition to OA, the physical interaction of atmosphere and ocean, coasts are open systems. There are several things that come in off the watersheds that can act to increase or decrease pH. On the other end of the systems are the ocean boundary conditions. For example, the upwelling of acidic deep ocean water is now a very well-studied phenomenon in the Pacific Northwest. Here in the southeast, South Atlantic Bight has a broad, shallow continental shelf, not so much of an issue here, although there's a lot of recent evidence for some complicated ocean currents that can impact pH locally and may change in the future. What I really want to stress is the ecosystem processes, the fact that biology is orders of magnitude more active in the coastal zone than in the middle of the ocean. And the processes of photosynthesis and respiration have a very direct effect on CO2 concentrations and associated carbonate chemistry. So as an example of pH variability in estuaries, I'll show you a time series from an estuary I know well, North Inlet. I'm looking out my window here at this system. We've got a long-term monitoring station in the middle of the basin. The reason I think it's a good example is that it's relatively undeveloped, minimal development in its watershed, relatively little freshwater inputs, complicating our measurement of pH in the system. And you can see we have about a 20-year time series, 30-minute observations, and unlike the trends I showed for the Sargasso Sea, we see a relatively flat signal, no long-term trend, but a surprising amount of variability on the short term, upwards of a full pH unit. Much of this is diurnal variability, day-night, on the order of greater than half a pH unit. And in fact, it's really clear if we dig down into this data that biology, that is photosynthesis and respiration, is the dominant driver of this pH variability. We can see that in this graph to the right, where the x-axis is the monthly mean daily range in oxygen saturation. Same thing for pH on the y-axis. So what this shows is that in the winter months, water temperatures are cold, biology is not very active. There's a relatively small range between the increase of oxygen during the day due to photosynthesis and then the decrease in oxygen at night as a result of respiration, and that produces relatively little daily variability in pH. But as the seasons warm up, water temperature increases, biology gets more active, production and respiration starts to ramp up. Now this daily swing in biology produces variability on a day-to-day basis, on average greater than 7/10 of a pH unit, which is really huge relative to that 1/10 of a pH unit shift that OA, the physical process, has contributed to date. I think what this means is that how production and respiration will change in the future really dramatically will impact our ability to predict pH changes. And certainly eutrophication has been a widespread problem for decades, is predicted to continue. This nutrient enrichment will very much change and influence production and respiration, influencing our ability to predict pH just due to ocean acidification impacts. I am going to skip that concept. I'm going to jump right to my last slide and talk about, so what does the future hold? Well, I'm not exactly sure. I think it's clear that there will be some level of future coastal eutrophication. There will be some level of further ocean acidification based on atmospheric CO2 emissions. Both of these will interact and they will come in concert with rising temperatures, higher sea levels, etc. So as a result, there's a great deal we don't really know yet regarding how all these changes will interact to alter future coastal ecosystem conditions. And I will leave the what these changes mean for the organisms themselves to the other members of the panel. I will end, I think I feel compelled to end with the statement that this lack of knowledge should not translate into a lack of concern. While we definitely need more work, there's a lot of uncertainty in our current state of knowledge. I think the precautionary principle is highly relevant here. Existing evidence suggests moving forward with strategies to mitigate climate change in general, of which ocean acidification is a part, is in our society's long-term interest. And I'm a little over, sorry about that. I'll stop there.
Thank you very much. So we'll now move to our next panelist, whose name is Mel Bell, who will be offering a fisheries and natural resource management perspective. Since 1983, Mel has served in a wide variety of roles as a marine fisheries biologist within the South Carolina Department of Natural Resources, but now holds the position of Director of the Office of Fisheries Management. In addition, in 2012, he was designated as the state agency appointed representative on the South Atlantic Fishery Management Council. So now I'm going to turn it over to Mel.
Thanks, Libby. Good to be here. What I'm going to do is what I'll call the 35,000 foot Mach 1 view of fisheries. I'm not an authority on ocean acidification, but what I'd like to do is basically present a picture of our fisheries in what we're calling the Carolinas, and what could be actually at stake or at risk, giving impacts potentially from ocean acidification. And let me say from the start that I'm not speaking for the state of North Carolina or the North Carolina Department of Marine Fisheries or the South Atlantic Marine Fisheries, the South Atlantic Fisheries Management Council. So I will just, this is my perspective on the fisheries here. So, okay, there we go. All right, so basically we talk about the Carolinas, we're talking about North Carolina and South Carolina, two states, but our fisheries are very similar in terms of makeup, not necessarily in terms of magnitude or size, as you'll see, but we basically target the same species that are important to us, and to keep in mind that our fisheries are basically commercial fisheries and recreational fisheries, both are which you'll see are extremely valuable. Commercial fisheries first. Basically, you can divide the commercial fisheries for both states into these major groups, both crustaceans, shellfish, and finfish. These are all very important for us, and as you'll see, related to putting a value on that, these are ACCSP data. This is an annual average value, ex-vessel value of our commercial landings for both states combined. As you'll see that the bottom, it's in excess of $100 million a year in product. That's the value of the product at the dock, if you will. In 2014, that was actually up to about 117 million. So it does move up and down. But you'll see that both of our states have about the same sort of proportion in terms of these major fisheries, whether it's crab, shrimp, finfish, or the shellfish. It's about proportionally the same. However, South Carolina is only about a quarter or less than a quarter of what North Carolina lands. Just another way of looking at value of this in terms of economic impact. These are NOAA data from 2011. But you'll see that once you start looking at economic impact of the commercial fisheries, North Carolina, you're pushing 800 million a year. South Carolina, we're an order of magnitude almost less, but it's still important in terms of the jobs created related to seafood industry. The industry does represent jobs. Jobs are important to the states, obviously. Now, the recreational fishery, we use the word recreational, and a lot of people, this is people pursuing the same resources that we're talking about commercially, but pursuing them for their own consumption or for just recreation or fun. But it is an extremely valuable component to our state's economy. So if you look at this, again, the NOAA data broken down by state. So in North Carolina, 4.74 million trips, individual fishing trips. I believe there are about close to 1.5 million licensed recreational anglers in North Carolina. South Carolina has about 478,000 licensed recreational anglers, but they generate a lot of trips. These trips generate sales of products, and you can see where you're talking in North Carolina, $1.96 billion in sales impacts associated with recreational fisheries. In South Carolina, scaled down. And then jobs associated with this fishery as well. It's not insignificant. So that's sort of the what is at risk potentially. There are significant fisheries, there are significant economics tied to those. So how does this tie into fisheries? In fisheries management, basically, what we focus on typically is fishing mortality. We regulate fishing mortality through size restrictions, seasonal restrictions, bag limits, those sorts of things. That's what we have control over. The factors that feed into a healthy biomass or healthy fish stocks, recruitment, growth, natural mortality, we don't have control over. But these are the things that ocean acidification or other environmental factors influence. And so to kind of put this in perspective, we are familiar with environmental impacts on fisheries. For example, changes in water temperature. When we get extremely cold winters, it impacts our shrimp crop here. In a short term, in a long term sense, changes in water temperature can affect the ranges of fishes. So we know this already. We're familiar with it. Salinity, another factor. When if you have either too much rain, too little rain, things can happen. We've experienced in the past couple of years oyster die-off from heavy rainfall, record-setting rainfalls. We've also seen the impacts of drought in a long-term sense on crab populations and abundance. Now, so ocean acidification, pH, another factor. This is where, and again, there are questions to be answered, but in the short term there could be there could be impacts on recruitment of, let's say, crustaceans, shrimp, crabs. If acidification could potentially impact the success of a particular year or years of these species. Long term, there could be the decreased quality viability of shellfish. So we're familiar with environmental impacts. pH could be just another one of these that we need to get our hands on. And in terms of what do we do about this? In terms of response, I think we need to continue or to increase regional field monitoring opportunities. I don't necessarily think we do as much field monitoring here as in other regions. That's something that could certainly be addressed. And also it may be that there are a lot of field opportunities or monitoring opportunities that are going on where we are collecting data now, we're just not necessarily pulling it all together. I do think we need to expand the laboratory-based physiology studies on species to see how species respond to differing pHs or differing scenarios for what might come down the line if things do change. That would give us a better understanding of impacts on recruitment, on growth, which are important. It would help us to be able to perhaps model and predict impacts on natural mortality. Remember back to that formula, natural mortality is we don't control that. We deal with fishing mortality, but if we can understand these other factors, perhaps we can get a better sense of what the impacts might be on our ability to extract from a stock. Also, this whole process would just help inform us to make better decisions in the future related to a management response, if there's going to be a management response. Another thing to keep in mind is that we're talking about the critters, but also we need to keep in mind the habitats. A lot of our habitats, we don't have reef-building corals in our area, but we do have habitats that are calcareous based, and if there are significant impacts on habitat, habitat impacts the viability of your fish stocks. And this is just point for me is that we deal in fisheries with a lot of competing needs in terms of research needs, data needs. So in gathering all these data related to potential impacts of ocean acidification, just keep in mind from a fisheries management standpoint, we have plenty of things that we need to do in the way of research, monitoring, and science-based fisheries management that we can hardly afford to do now. So we have a long laundry list of unfunded or not particularly well-funded areas that we deal with constantly. So, don't want to see this initiatives kind of becoming competitive in that, just from my perspective where I sit in fisheries management. And that's it for me. I guess we won't deal with questions now. We'll deal with that later.
Thank you, Mel. And on that note of questions, if questions do present themselves to you all as the audience, make sure that you type them into the chat box so that we can, or the questions box so that we can pose those to our panelists. So now we'll move to our third panelist. Our third panelist is James Morris, an ecologist with NOAA's National Centers for Coastal Ocean Science, which is within NOAA's Ocean Service Division. James hails from a sixth-generation coastal Carolina fishing family, hopefully I characterized that right, James, which has been involved in shellfish aquaculture for the past 30 years. He will be sharing insights both as a result of his family's background and his current position in NOAA, leading the Ocean Service's aquaculture efforts. So I'll turn it over to you, James.
Thank you, Lindy, and I apologize, folks, that I don't have a set of slides for you, and honestly, I feel like the least qualified person to really be on the panel. We have not been working programmatically on any ocean acidification projects here in our laboratory in the context of aquaculture and fisheries stuff. Although one of our recently retired employees has done a bit of ocean acidification work, which I'll share a little bit about in a second. This issue, though, is one that has been on the front of our concern, especially from an aquaculture perspective in the southeast region, given what we have observed in the Northwest. And I'm sure that Bob will later perhaps talk a little bit about that. I think that there's a lot of uncertainty and a lot of concern moving forward in terms of the interactions between ocean acidification and the other coastal environmental stressors that we deal with every day. It's probably the interaction that concerns me the most as we move forward. I would like, though, for just a second to hit on a couple of things and to reemphasize some of the points in the earlier presentations. One is that the Carolinas are have such a unique system. We have recently a retiree here gave a presentation last year at an event, and I scribbled down some notes from that. And he presented some data to show that how diverse the fisheries are of the Carolinas, and particularly in North Carolina because of the size of the estuaries that we have and the mixing zone that occurs here at this natural biogeographical province that we call the Carolinian province. He provided data dating back to some of the work that Frank Schwartz had done at UNC and others in the region that quantified the number of saltwater fish species that exist in the states of Virginia, South Carolina, and North Carolina. And he showed that there's approximately 350 saltwater fish species in Virginia. There's approximately 450 saltwater species in South Carolina. But if you enumerate the number in North Carolina, that number is up around 685. And I was really astounded to just having grown up here and worked in our commercial fisheries and just being amazed by the biodiversity that exists in North Carolina. I was really impressed about just how biodiverse this region and this state is. In addition to the uniqueness of our fairly closed, high residence time, very large estuaries, sounds and its estuaries, and what a unique place it is to study a problem like ocean acidification. I've also been impressed that we've seen a decline in the last several decades of our commercial fisheries, and there are lots of other interactions that are happening here that affect our fisheries and aquaculture industries. We've heard a lot about eutrophication, rising temperature, salinity, habitat loss, the overfishing, and a lot more. And it's very difficult for ecologists to tease out cause and effect from one specific parameter on any type of those stressors, and that's certainly adds to the challenge of this problem as well. A few points, though, that I would like to highlight about ocean acidification, and some of these have been highlighted, so I'd like to reinforce some of those. One is that, as we saw in the Northwest, the shellfish aquaculture industry can provide really an early warning system. From firsthand experience, the industry is having a really rough go right now in terms of hatchery production of shellfish, clams and oysters and scallops in the southeast region. We are seeing, in the last few years, we've seen significant reductions in hatchery production coming from the region, and we're not sure why. There's a million different theories and possibilities, but it certainly raises alarm that are we dealing with changing ocean chemistry? Are we dealing with changing influences of agricultural runoff or coastal development and water quality issues? What are these challenges there, and what are the resources that we have to address some of those things? But I'd like to just highlight what a critical role the shellfish industry, and particularly the hatcheries along the East Coast, and particularly in the Southeast region, that working with them as sentinel sites and ears on the ground that are working with coastal ocean chemistry and early life history of bivalves can provide really important data and information that can inform this process. The other thing I'd like to point out is that very early here at the Berkeley Laboratory, we began a course of study on the important role that seagrass, particularly submerged aquatic vegetation, has. And we've had a few folks now that have left this laboratory that impressed upon me the importance that SAV has as a buffering and mitigating influence in estuaries. And there was, back a few years ago when there was a lot of concern about acidification, and I was really asking a lot about this problem, I was reassured that our seagrass biomass would help in buffering those ocean chemistry changes that would happen. And I wonder about that today, and I wonder about the biomass that we have and the masses and the mass amount that we have in these coastal estuaries. What is the influence that it's having? And we know from the chemistry equations that it's possible, but measuring that and understanding its role on an estuary level and an ecosystem level is something that begs more work. Another thing is, and it was mentioned in the first presentation, but Dr. Bill Sunda here, recently retired, but has spent a career working on ocean chemistry issues, and particularly in cycling of carbon in the ocean. Really did some phenomenal work looking at the interactions between eutrophication and ocean acidification. And we can't talk enough about the importance of thinking about the two in concert. And I reinforce the presentation that was given earlier, in that we really have to consider both of those issues and how they interact. And I understand from Bill's work that interaction is not necessarily one that is linear, that we can that we have to be concerned about at some point reaching a scenario where the impacts become much more higher in magnitude. And then lastly, we do a lot of ecological physiology work around aquaculture, around fisheries here at our laboratory. And I certainly get most alarmed when I think about the interactions that ocean acidification can have on the behavior and the early life history of our coastal flora and fauna. And we have a lot of work to do in this temperature regime. There's been a lot of work done in the cooler temperatures of the Northeast and Northwest, but I believe there's a lot of work yet to do in this region. And I look forward to hearing the discussion and learning more from you and the questions that are being asked, and contributing to this discussion. Thank you.
Thank you so much, James. And so last but not least, Bob Rowe will be offering the industry perspective. Bob started his aquaculture career in 1986 when he built a small shellfish hatchery in Rhode Island while he was working on his PhD. He's done international consulting work on hatchery design and algal culture, and he now serves as the executive director of the East Coast Shellfish Growers Association. So I'll turn it over to you, Bob. And we are not hearing you, Bob, so I think you need to unmute.
Sorry about that. Libby, appreciate the introduction and the introduction of all the other speakers before me. I'm going to try and be real brief. I don't know that I've ever been able to say anything in 7 minutes, but I'm going to blast through some stuff real quick. The East Coast shellfish aquaculture industry is about a $156 million industry. Virginia is the biggest player, but we've got pretty well spread, but the Southeast is pretty well challenged. Just going to give you a quick official disclaimer. These are my opinions, not those of my association. When we first heard from Richard Feely about 6 or 7 years ago about ocean acidification, we initially panicked, and now I don't know what to think, quite frankly. I am certainly not a climate change denier. This is very simple chemistry. We are seeing the impacts of the additions of CO2, but what we fail to do, I think, and the point of my talk is we haven't really well defined how this is going to affect the biology of the organisms. Frankly, we wouldn't care about the pH of the oceans if it didn't have an impact on the fisheries and the animals that we raise. So we have lobbied heavily for the 4M Act as an association, and somewhat to my dismay, a large amount of this funding seems to be spent on oceanographic cruises and monitoring, while frustratingly little has been spent on figuring out how this is going to impact our organisms. I just want to repeat what Eric said, that it really doesn't serve us well to conflate these issues of upwelling with eutrophication-induced acidification and the real issue that we're talking about, which is ocean acidification, upwelling. Apologize for that, but anthropogenic sources of CO2. We need to know how this is going to impact our fisheries, and frankly, a lot of the science that has been done, which involves just bubbling CO2 into a beaker, is sort of a weak proxy for the real-world conditions. Eutrophication-induced acidification is happening, and we are seeing very high levels in many of our eutrophic estuaries. These are levels that we are predicting for 100 years from now. We have seen declines in shellfish biomass, but there are still successful sets in most of these estuaries. We see this huge normal daily variation in pH, sometimes as much as a pH-and-a-half unit swing. And so, you know, the question is so why should we worry about 1 or 2 tenths of a pH unit? Well, in many regards, I want to caution you that this is a specious argument. We see temperatures go up 20 or 40 degrees a day. Why should we care about a half a degree of global warming? Well, this has had tremendous impacts on our fisheries with organisms moving northward at 1 kilometer a year, and significant changes in sea level and other impacts, storms, rainfall. So I don't want anyone to think that I'm sanguine about this, but I just want to point out that we do have these huge changes. Also, that shellfish have evolved through a tremendous amount of climate change, and that, you know, okay, just to back up a little bit, there is a huge amount of genetic variation in these species, and they may tolerate the low aragonite saturation state that is projected for the next 150 years. But we're not sure, and the science is quite uncertain. We've seen the Sydney rock oyster adapt, I'll use that in quotations because I believe that's epigenetics, but we've seen that they are more resistant to low pH conditions. We've seen that the Gigas reared from adults raised in low pH conditions are more resistant to the acidic conditions. We just don't really have a good feel for it. This was a study that was done in 1996, and you can see over a pH range of 6.5 to about 9. Survival rates of oyster and clam larvae were up over 90%. Now, I don't want to throw Tony Calabrese, who's one of the titans of our industry, under the bus, but I don't know that I ever had 90% survival in a hatchery. Here's another interesting study that was done by Gary Wickfors and associates up in Milford, and if you look at the internal pH of the cell that is actually laying down calcium carbonate, constructing the shell, it is retained even though the animal is plunked into 6.7 pH water. The cells that lay down the shell have very active proton pumps, and they are able to retain that low pH, considerably lower than the pH of the outside seawater, I might note, even before they were put into the 6.7 pH water. Meanwhile, the hemolymph is conforming to the pH of the water that it's been put in. The carbon isotope ratio of the shell is indicative of the food rather than the ambient water. So maybe our fascination with aragonite saturation is misplaced, but we just don't really know. So we know it's coming. We know it's coming along with sea level rise and eutrophication and hypoxia and a lot of other challenges. But we really don't have a feel for how this is going to impact shellfish growth and recruitment. We do know that larvae are extremely susceptible for the first 48 hours. We also know that hatcheries are able to get us through that very sensitive phase with carbonate additions. Everything that I've read suggests that aragonite-based corals are probably headed for extinction, although I just read a story about a Palau reef that has CO2 bubbling up in 1,000 ppm CO2, and they have excellent biodiversity, and the reef seems quite healthy. The scariest thing in my mind is that we can't yet rule out a collapse of the marine food chain. Salps and coccolithophorans form the base of the marine food chain, and should we have a collapse of that marine food chain, it would be a global calamity. And then, these nitrate levels and the eutrophication that we see in many of our eutrophied estuaries do present a clear and present danger. The acidified muds that we see in many of our areas are totally inhospitable to larvae, and we do see reductions in biomass of certain species of shellfish now. So I'm not suggesting that we have nothing to worry about. I just am complaining that we don't have enough information. I'm pleading for more research money. I need to know how this is going to impact my organisms before I'm willing to try and answer the question of how it's going to impact my industry. The last time we had levels of CO2 of what we're predicting for the next 50 to 100 years was in the Paleocene-Eocene boundary, and there were mass extinctions. And the fact that we're even mentioning the word mass extinctions should give everyone great pause. And that happened on a time scale of 50,000 to 100,000 years. And we're predicting these changes to happen on the scale of 50 to 100 years. So can shellfish adapt, or will there be mass extinctions? Probably some of both. And that's my message. I need some more quality research to be done. Thank you.
Thank you, Bob. So that's the end of our panelist presentations. Thank you all so much for offering your expertise and perspectives. We'll now move on to questions for the panelists. And again, want to encourage all attendees to also ask questions. And I do see that a few have come in, so we will be getting to those shortly. But first of all, I'm wondering whether any of the panelists have any questions for any of the other panelists.
Libby, this is Eric. I'd like to just follow up with Bob's last statement for plea for more research, and because I loved his earlier comment about bubbling CO2 in a beaker being a poor proxy. So Bob, I'm wondering if you can elaborate or provide a perspective on what kind of additional laboratory, or perhaps more importantly field studies, are needed to assess effects of OA on the biota,
Sure.
at sort of the population to community level, and get away from the beaker experiments.
Sure. Absolutely, Eric. I think that some mesocosm work, I think we need to have sediments involved. I think we need to have algae involved to suck up some of that CO2. We need to have diel cycles like we have in nature. And then I'm willing to accept that represents more of the real world. I really question some of the very simplistic experiments that have been done without plankton, without sediments. It just leads to a huge question in my mind as to whether these are really predictive.
Yeah, and perhaps without, substantial oyster reefs that provide their own inherent buffering capacity. I like James' comment about the teasing apart cause and effect when there are multiple interactions going on.
It's certainly harder to do good science when you can't keep all of the other variables constant.
Yeah.
So Bob, Libby here. So just following up on your point, I would point you to the important work that our European colleagues have done, particularly Ulf Ribesal, who's had those gigantic mesocosms that include all of the factors that you've spoken about, and papers are constantly coming out with results from those studies. Just want to.
I look forward to seeing them.
Mm-hmm. So do any of the rest of you want to answer Eric's question? Otherwise, we'll move on to another question. Sounds like we're... So a follow-up question for Eric from me is, in your opinion, is there a need for enhanced monitoring in the coastal zone, and do you have ideas about where more specifically this monitoring should take place and on what time scales?
Yes, I have opinions. Well, so I think, first of all, if we're talking about the Southeast, there really is currently an almost complete lack of OA monitoring infrastructure in our waters. I think Gray's Reef is the only true OA observatory, and while I showed a long time series of pH in North Inlet, that's the old school pH sensor. I glossed over caveats to that. Monitoring just one component of the carbonate cycle is insufficient. You need at least two. I mean, we really need to get at aragonite saturation levels. Going back to James' comment about the need to tease out variability when you have multiple effects going on, I think that infrastructure is almost entirely lacking in the southeast, in the inshore, which is where I think we need it when we talk about the interaction between eutrophication and ocean acidification, and what that means in our region that is, got some of the highest population growth, coastal development rates, is poised for much further eutrophication pressures. I would also address that question to Mel and some of the others. Well, Bob's right. We care about this because of impacts on the organisms. So where is that information most critical from the standpoint of the fisheries management? I'm assuming that's inshore, estuaries being the nursery ground for most of these commercially important organisms, but to some extent I would defer to folks like Mel to tell us where we need those monitoring capabilities.
Mel, would you like to follow up?
Yeah. Well, sure. And again, it's not my area of expertise, but in terms of the monitoring, when we're talking about monitoring, are we talking about actually monitoring the pH levels at different sites? So some of that may be occurring offshore related to, for instance, our MARMAP program, Marine Monitoring Assessment Program, that visits stations offshore on a regular basis, not all the same stations every trip, but they do collect and have over time collected water samples and taken parameters such. Salinity, pH is one of them. I think since 2011 they have pH data. Now I'm not familiar with exactly how they're doing that. So there is a capability of getting perhaps some offshore monitoring in place by relying on programs that are already out there taking water and looking. MARMAP, CMAP, there may be others as well. So that's offshore. Then as far as inshore goes, I know, Eric, you've got your station up there in the near, perhaps the near down south in the ACE may have something like that going on as well. But obviously our fisheries that we talked about are extremely tied to the estuaries. So any changes in these estuarine environments related to pH could have a tremendous impact, whether it's shellfish or whether it's shrimp, crabs. I mean, you name it. Even offshore species that we think of as offshore species, where we harvest them, have a connection to the estuaries in terms of their where they grow up. So I would think that there could be, and again, it's not my area, but some standardized stations set up to just monitor what's going on throughout the different estuarine systems. And that would be really expensive. But offshore, we do have the capability of getting some data now.
I guess I would just add the caveat that what we've seen so far, high frequency, Libby, you had a part of your question that was the temporal resolution of measurements. Clearly the variability in pH in the coastal zone has a very strong tidal, diurnal, seasonal signal to it that continuous high frequency measurements is pretty critical to understanding that variability.
Okay, so I'm going to ask, I have one more question, then I'll turn to the questions coming from our audience, since we have a few of those. So I guess for either Mel or James or anyone who wants to answer it, given the potential vulnerability and high economic value of commercial and recreational fishery species in the Carolinas, I'm curious if you know whether either state is considering OA and the management plans for those coastal species. And I'll just say, for instance, the governor of Mississippi recently convened an oyster council, which concluded that OA is one of the several important factors which needs to be taken into account and monitored as they restore their oyster industry. So Bob may have answers to that as well, but maybe start with Mel.
Yeah, and I checked with one of my colleagues in North Carolina briefly, and so I think the answer is, well, not exactly. It's not a factor that has been incorporated directly into any management plans. North Carolina, as a state, operates through management plans for various fisheries up there. We operate a little differently here, but there's ocean acidification by itself has not necessarily been identified as a specific item in any management plans that I'm aware of. There's obviously within the shellfish community in North Carolina and here as well, there's a lot of interest in this. So you may see it come up mentioned a lot more in the context of dealing with shellfish resources, but to my knowledge, it's not really incorporated. And I know from the council perspective in looking at federal fisheries, it's not something that we spend a lot of time talking about, or it has a specific little component of any plans that I'm aware of. That's just from my knowledge.
James, you don't have any information about that, do you? I'm not sure if you're muted.
Yeah, I just unmuted. I'm not the best one to speak to this. I know certainly that it's a consideration in various management planning processes, but I am not aware of any management plan that specifically lays out any course of actions on ocean acidification, whether it be developing monitoring networks or calling for specific research, as Bob alluded to here in our state. But again, I haven't researched this, and I certainly can't speak for our state. I'm not aware of any federal programmatic work along these lines in the region in which is funded annually, whether it be monitoring or not here. So it's definitely something that could possibly be put down as a possible action item moving forward.
Mm-hmm. Okay, so I'm now going to move to some questions that have come in from the audience. The first one from Dan Martino, and he asks, how well does the calcium release from dead shell, that is from oyster shell, combat the rise in acidification in the water body? Is dumping shell into a water body a good way to help keep the pH stabilized? And I think maybe Bob would want to take a first stab at this.
So early on in my career, I was growing oysters in black and oxtic mayonnaise and had a mass mortality and dumped out several $100,000 worth of driveway onto my lease and proceeded to catch a set of hard clams, unlike any that had ever been experienced in this particular area, so dense that they were pushing each other out of the sediment. So yes, we can sweeten the bottom with shell, especially these highly eutrophied muds, deeply acidic muds that are totally inhospitable to shellfish larvae. There's also been some work by Mark Green up in Maine looking at potentially rejuvenating some of these flats that are suffering. Maya production is down, and one of the concerns may be that it may be eutrophied muds. The rate of dissolution is quite slow. It's much faster when you have these acidified muds, but there was a gentleman in my state who claimed that he would go out and throw pelletized limestone out on the beds to catch a good set, and this was 30, 40 years ago. So yeah, sweetening the sediments is a possibility.
And a follow-up question from him is, how can shellfish farmers help with the research? So that may be to either you or James.
I'm sorry, I missed the question.
Sorry. So Dan's question was, how can shellfish farmers help with research on ocean acidification?
Well, I always try and get my members to participate in research, assisting when a researcher needs a field site, things like that. But I believe that we need some really solid lab work done here by some really top-notch researchers.
Mm-hmm. James, do you have any?
Yeah, I'd like to mention something along those lines. I'm a big proponent and supporter of extension, and I see Julie Davis is on the line here. We have a new aquaculture extension specialist with Sea Grant, Chuck Wyrick, here in our state. And I just want to emphasize the importance of those extension folks in being on the ground and working with the various growers and hatchery managers and such, and working to keep keep awareness high and collecting long-term information about the performance and productivity of the industry, and being able to work to continue to tease out. Growing shellfish is more of an art than a science, and we all realize that. But at some point, you do reach you do have enough evidence to be able to point you in the right direction. The increasing need for extension folks to be on the ground and be working with growers and identifying questions and ruling out problems and clearing those things is absolutely critical.
Yeah, I would add that I think that we're going to see the most impacts in the hatcheries, and for those hatcheries that have been able to work with researchers and install the appropriate monitoring equipment, such as Bill Mook up in Maine and some of the guys on the West Coast, they are able to mitigate the challenge by carbonate additions. And I think that we're going to get some real good, much better understanding about the interactions between aragonite saturation and larval survival over the next couple of years as more hatcheries are able to install this kind of machinery. If it was just pH, it would be very simple, $60 meter, but it's not. It's rather complicated getting at the aragonite saturation.
Mm-hmm. So we're close to running out of time. I'm going to pose one more question. So this is from Lou Burnett, and he asks, CO2 in coastal waters currently exists at 20 to 40 times the highest projected end-of-century predictions. Water pH fluctuates with CO2, and pH can fall to values below 6.0. Slight changes in atmospheric CO2 are unlikely to affect significantly acid-base chemistry in local waters, and yet fisheries continue to exist. Oysters, whose larvae are very sensitive to pH, continue to settle and grow in nature. How is this possible? So maybe Bob or Eric?
I mean, as I said in my talk, I don't think that we can be sanguine about the change of a few tenths. 1/10 of a pH unit represents a 30% increase in hydrogen ion. It's a logarithmic scale. And for the same reason that, half a degree of temperature increase in the last 50 years has caused mass migrations of finfish and lobsters and had tremendous impacts on storms and things like that. I don't think that we can be sanguine about this. It begs the question. We need to know how this is going to impact our biota, and I don't think we have the answer yet.
Mm-hmm. Thank you. So we're going to conclude our the SOCAN webinar. If we did not get to your questions about the content of today's presentation, feel free to contact our panelists via email. You can find their email addresses in the chat box in the control panel on the right-hand side of your screen. If you're interested in joining the SOCAN listserv to learn about future updates in the Acidification Network, please sign up at the URL shown on this slide. And there it goes. Please direct any questions about SOCAN to socan@secoora.org. Thank you again for attending the SOCAN State of the Science virtual panel today. And thank you very much to all of our panelists. I think were provided some excellent background and perspectives. We welcome any feedback, further questions, or suggestions for topics for this webinar series. You can submit input by replying to the follow-up email you receive. The recording of this webinar and a PDF of the presentations will be available on the SOCAN website shortly. Our next webinar will focus on the state of the science here in the Southeast. We will have Astrid Schnetzer of North Carolina State University presenting on understanding ocean acidification and the microbial community on Tuesday, September 8th at noon. Registration for the webinar and where to send those questions is now available on the SOCAN website. Thanks again to all of our panelists and to all of our audience for the very thoughtful questions. I'm sorry that we didn't get to all of them. And thank you for taking the time to join in. We hope to see you at the upcoming State of the Science discussions. And that's a wrap.